Hello, my name is Eva Dyer, and I'm a professor at Georgia Tech and Emory University. Today, I will tell you about exciting new work from my lab in being able to leverage new tools that are being used in different areas of machine learning called self-supervised learning, and leveraging these self-supervised learning techniques to build representations of neural activity. All right. So I'll start off just by acknowledging the many individuals across many institutions that made this work possible. My folks in the NERDS lab at Georgia Tech on the left. And this work that I'll describe was really spearheaded by Mehdi Azabu, as well as our collaborators at WashU St. Louis and at DeepMind and Johns Hopkins APL. And then I'd also like to acknowledge our sponsors, including NIH and the Theory, Methods, and Models program. All right, so the underlying motivation of the work that I'll describe today is driven by, a pervasive question in neuroscience that is, given some observations of many neurons in the brain, how can we take that information, how can we take those measurements and build meaningful representations of neural activity that allow us to get a lens into the underlying behavior or task or intent that the animal or individual is, sort of representing in their brain. And this is a really hard problem in general. There's many different approaches that have been taken to build representations. But with that, we still are facing a number of challenges that have to do with the stability and the sort of utility of these representations. And often neural states or activity patterns of the brain will actually shift over time, or when we come in and record at a new day, we'll have access to a different subset of neurons. And so these different sources of variability do make it hard to build a representation and then use it and carry it forward in, say, brain-machine interfaces or other downstream applications. So we were really interested in whether or not we could use new tools that are really being established in computer vision and graphs and other areas of machine learning. We were interested in whether or not we could leverage these new state-of-the-art tools in a neural domain in order to solve the problem that I described, which is building representations of neural activity. And so, the first question, or just I'll start by describing what self-supervised learning is and what are the principles underlying it, and then from there kind of describe how we can use these same general principle ideas and apply them within neuroscience. So first, what are the principles underlying self-supervised learning? Unlike other methods for building representations that rely on essentially reconstruction or generating patterns of activity that resemble the inputs, the idea behind SSL, self-supervised learning, I'll just call it SSL, is to essentially take an input. So in our case, we'll think about a collection of activity of neurons, and that will represent a brain state. But what I'm showing here as just a sort of illustrative example is, a case where we have an image. And from that sample or that image, the idea is that we'll basically generate through some transformations two different views of that same underlying sample or image. And what I'm showing here on the right is a kind of demonstration of this. So, we have a picture of a cat, and then we're applying two different transformations to create different views of the same cat, right? Either changing the color or in the bottom example, zooming in, and cropping so that we have a different view of the same cat, as I said. And so the idea is to basically take those different views and then learn a representation, often with a neural network, that basically is trying to maximize the similarity or minimize the distance between the embedding of these different views in the latent space of the neural network. And so as I outline here, the approach is create augmentations and then pass them through and try to maximize similarity. And really the intuition behind that is if we give the neural network really diverse views of something with the same underlying semantics and we can learn a representation in this way, then we can basically get rid of all of the other nuisance variables or other sources of variability that aren't essential to the underlying task, which in this case would be to recognize cats from dogs or other images. And so the set of transformations or augmentations that we apply, we will build in different types of invariants into the representation. So, you know, as I sort of alluded to, self-supervised learning methods are, you know, providing very impressive performance on a number of complex tasks in natural images and in computer vision. And now, over the past year, these methods have started to actually approach the performance of supervised methods that are trained end-to-end. Whereas, you know, in this case, as I described it, we do need some information about the augmentations that we apply, but the actual method itself is a fully unsupervised representation learning method. And so this is an exciting new advance in the field to show that unsupervised approaches can really provide these rich features that will rival the performance of supervised methods. So a core question that we have to ask, right, is how are these views of a sample generated, right? Because that will be critical to the sorts of representations that we build. And so in vision, as I sort of said before, the intuition is that we're interested in applying some transformation that will still keep the essence of the cat or the dog in this case, so not changing the semantics, but providing these, you know, really diverse pictures of the same object. And in images, we know of really rich augmentations or transformation classes that will solve this task, right, of capturing underlying semantics while also providing pixel level content that's, you know, very different. And so when you have these rich augmentation classes, for instance, in vision, you can, as I showed before, achieve these really state-of-the-art performance, right? When you have rich enough augmentations that you're building in, but in new domains, really understanding what the right augmentations to apply are is challenging, and it's not very well characterized yet. And so when we set out to, you know, think about how these ideas could be applied within neuroscience, we started with some kind of, you know, core principles that are guided, you know, as the topic of this workshop, guided by theory. And in particular, we sort of look to or have insights from predictive coding models, which have been leveraged extensively in neuroscience. And so the first idea is, okay, we want to find something that is that should have shared or similar semantics to the brain state or the sample that we're interested in augmenting. And so how we can do that is first by using just a sort of temporal consistency, or the fact that if I'm, you know, as I'm showing here in the picture, so X would be the activity or the firing rate of multiple neurons stacked into a vector, and that's now the brain state vector or observation that we're interested in. And then when we build a sort of augmentation, what we're interested in doing is figuring out the common or the shared information between two neighboring points in time, right? And so X and X prime could be just defined as either, you know, some forward steps or backward steps or some random view that's within a local window in time. And so that gives us at least an initial idea of how we might be able to map this idea of information maximization-based learning into a neural framework. But we were interested in whether or not we could actually go further than just looking very local in time, right? Because ideally, because we don't know the right transformations to apply to create augmentations, ideally we might be able to look from within the data set directly to figure out what are the sources of variability that we need to build invariants to. And as I described at the beginning, one of the challenges that we find is that neural activity patterns do drift. So being able to link and learn representations across non-local or, kind of temporally distinct points in time would also be a really powerful strategy. So really the goal is to build a framework where we can leverage the diversity within our neural activity or within our data to solve this augmentation or self-supervised learning task. So as I said, first we'll look locally, and then as we start to build a representation that is sort of stable in time, really our goal is to go beyond just those local views and to start to group similar experiences or similar brain states that might not be close in time, but should have shared or similar semantics. And so in order to achieve this sort of this goal, right, of having both this predictive coding framework that works on a local but also non-local scale, we developed a new self-supervised learning approach that's general, so can be applied to images, and we're also interested in applying it now to graphs, but as I described, was really motivated and driven by this neuroscientific application and this need to create diverse augmentations of data when we otherwise might not know how to apply them directly. So is called Mind Your Own View, or MEOW, and we build this framework on top of a sort of existing predictive engine that I showed at the beginning that's really achieving these SOTA or state-of-the-art performance in ML, but none of these methods had been applied within neural activity or neural populations. And so we start with this baseline framework that I showed at the beginning, where we, can create augmentations through these transformations, pass it through a network, and then essentially try to minimize a normalized Euclidean distance between these different transform views. And then in addition to that, in addition to this term, which in our neural example works over these local and dropped out views of neural activity, so by masking certain neurons and jittering locally in time. And then in addition to that, we build a sort of cascaded architecture that starts with those local predictions and then adds in an additional branch for these non-local predictions, or in the case of images, we would actually look from within the data set, find another candidate view that is similar in the representation space in the network, and then use that then as a positive predictive target for this self-supervised method. And so here on the bottom, I'm just showing an example of this, where we're passing in the same image of a cat. We can transform it or look locally to create an augmentation, and then we'll basically try to look over a set of other images or other samples, find the closest one or a near neighbor in the representation space of the network, select that, and then pass it through the network in a similar way to maximize similarity. But now, instead of having other examples of the same cat, we're leveraging the fact that there are other pictures of other cats that are similar to kind of build a more unified perspective on this. And our loss is just a mixture of this local and non-local sort of term, which I'm showing here over on the right. And you can trade off the sort of amount of local versus non-local. Okay, so that's the sort of overview of this general approach for self-supervised learning. And now I will dig into the results and the application of this idea in two different settings in neuroscience. And so the first one is a slightly simpler task, or a task where an animal is trained to make reaching movements, targets, there's basically a cue that comes on the screen and they'll make a reach. So standard center out reaching task, we're recording from neurons in the primary motor cortex in this case, roughly about 100 to 200 neurons across these different data sets. And what I'm showing here are the results of essentially training this neural network using this local, non-local strategy in order to find representations and then comparing with other baselines. And what we do is we train the network in an unsupervised way. We basically freeze the weights, and then the goal is to use the representations to just form a linear readout, so only a single linear layer that's appended to the representation to read out some decoding information. So we're using this decoding task as a way of understanding how well the reach direction in this case is already being embedded within the representations of the neural activity. So each group of bars is a different data set. We have 2 different days from 2 individuals, so Chui and Mihi, and comparing with an autoencoder and SimClear, Bio, so other SSL methods. And so the take-home message here would be that, you know, not only does Meow, the proposed approach that uses a combination of local and non-local augmentations, you know, provide a significant improvement over these other baseline approaches. The other thing to take away is that if we look at the supervised performance, so the accuracy that we get when we test on a held out chunk of time on a supervised decoder trained on this same task, what we find is that actually the SSL approaches will often outperform a supervised decoder, which is a really exciting result because it shows us that there are important forms of regularization and you know, information that we're embedding through these augmentations that allow a neural network to perform better at test time on held out data. So we're forming, in some sense, more stable representations that allow us to decode this particular behavior effectively. We can also look at the latent spaces or sort of visualize the representations that are being formed in this case. And we find consistently that by using our strategy, we can preserve more of the global structure of the task. So here, showing results from Meow after applying t-SNE in this case to find some low dimensional embeddings. And here we're finding that the kind of the circular nature of the task is being preserved within the representation space, whereas we don't see that with some of the other competing methods. Here I'm just sort of showing a number of the ablations that we provide in the paper, which is out on arXiv, to kind of explore how incorporating different sources of noise, or as I talked about before, different amounts of dropout, right? How does that dropout or the temporal jitter augmentations that I described, how does that impact the overall performance in terms of first building this stable and good representation? And what we find is that consistently, both a combination of dropout and this sort of local prediction that I described is, both of those things are essential for building meaningful biases or regularization into the network that allows us to then really leverage this non-local structure further with Meow. And these are just now kind of zooming or looking at the representations of the data in 3 dimensions and showing that we can both preserve the different reach directions, which I'm showing here over on the left, where all the different points are color-coded by the actual reach direction. We're showing nice separability. But in addition, over on the right, I'm just showing that the The other aspect of this task, which is moving from a lower to high velocity in this outer portion of the latent space, both of these aspects of the behavior are being captured in these neural representations formed by meow. Okay, so after establishing the method on a number of different data sets in the macaque, we then went on to apply our approach to free behavior. So in this case, we have recordings from different rodents. I'll mainly talk about, well, I have an example from the mouse hippocampus on the right, and also from rat V1. And we're trying to expand this into even more diverse brain regions and really understand how well these sorts of ideas generalize to different sorts of behavior in different brain areas. And so what I'm showing here now is we apply approach, and now we just read out through this linear layer. We're going to read out information about sleep state. So here we're trying to decode REM from non-REM from wake across large 12-hour blocks of data. So this is while the animal is running around in their cage, and there's no explicit, excuse me, no explicit training. It's just free behavior. And so what we find, or what we can, say, is that dropout as an augmentation continues to be a powerful form of regularization. And we find that our results overall hold in terms of, either outperforming or matching a supervised decoder on this sleep task where we only have 3 classes. And so it's a bit of an easier readout. And, really just further confirming that these ideas can be applied in more diverse domains. What's interesting is that even though our behavioral labels are relatively coarse, right, being awake or being asleep, if we actually look at the underlying representations that are being formed by meow in comparison to, say, just using the raw firing rates or the raw data, even though the behavior is nicely separable, we get very high accuracies, we actually find that there's a lot more going on under the hood, a lot of interesting sort of clustering and differences in the representations. And so we are interested in digging into that further. And in the last few minutes, I'll just describe some of our preliminary results, which we're really excited about because we see that, this tool gives us a way of finding unique parts of the representation space that seem to be linked to unique and complex behaviors. So just, without being able to go into this into too much depth and show all you, we developed a tool that's sort of just an extension on TensorBoard where we can take the representations learned by the network, zoom in on them, and then get additional behavioral readouts and see the position, whether or not meow is finding other points over distant points or over I guess what I'm trying to describe it here is the plot. I can't show with my cursor, but on the upper right-hand side where it says selected cluster. And so basically the black lines show all of the points in time that the network is sort of saying are very similar in terms of the underlying brain state that was present. And so we do find that there are, local points in time, of course, where the animal is doing similar things, but then the network will also link distant points in time. And when we go in and confirm, we find that often the animal and the behavior is quite similar. So in this case, just looking at an example where we have water drinking on the bottom are insets of the video, finding clusters in the latent space that correspond related to nest building or, kind of getting comfortable but not sleeping, so a quiet wake state, being more active and playful and moving their box and other objects. and so on and so forth. So we're starting to now be able to leverage the unique insights provided by self-supervised methods and our approach in particular, Meow, and use that with these really large untethered data sets to build insights into encoding, in this case in hippocampus, that we're really excited about, describing in terms of the neuroscientific implications as well. All right, and before I end, I'll just kind of quickly give a plug for some related work where we've been able to take this idea of invariance-based learning with self-supervised learning through Meow and kind of combine that with a generative modeling approach or a variational autoencoder. And this is recent work. I have references here if you're interested, called Swap-VAE, which essentially is combining these self-supervised methods for neural activity along with a VAE to kind of hopefully get the best of both, where you can see the things that are, kind of stable and common across local views, while also having a full range of variability necessary to actually generate realistic neural activity. And we are using this to understand and try to build disentangled representations of neural activity. All right, so I'll go ahead and conclude. So in summary, I described this new approach for SSL called Mind Your Own View. I didn't describe the results in natural images, but we're finding that we can compete with state-of-the-art methods in CFAR and Tiny ImageNet and some of these other computer vision data sets. We described specifically the application of Meow to neural population activities, discussed what it means to form augmentations of neural activity and show that those work in both rodent and macaque. And then also demonstrating that a cross-sample prediction with Meow often beats the supervised baseline. And finally, just a little preview of some work that we're doing where we're trying to combine these ideas with generative modeling approaches and showing that that can be used to sort of disentangle things like reach direction from the actual dynamics. So with that, I will thank you so much for your attention and invite you to check out some of the code or some of the other work that I presented on either of the two links. Thank you so much.